MSc degree from Kenyatta University in 1988 and B.Ed Science from the University of Nairobi in 1985. In 2010, he attained a Master of Arts in Leadership, MAL, from Pan-African Christian Park University. He first joined the Department of Chemistry, Kenyatta University, in 1988 as a graduate assistant and has grown in the ranks to a professor. He is involved in teaching both undergraduate and graduate programs, supervising graduate students, and providing academic leadership in the Department of Chemistry and in the School of Pure and Applied Sciences. Professor Nyambaka is also the chairman of the Department of Chemistry from 2012 to 2016. Professor Nyambaka is a member of various chemical societies, including the Kenya Chemical Society and the Royal Society of Chemistry, RSC. He received scholarship awards from Kenyatta University towards his Master of Science degree, that's from 1985 to 1988, and the British Council towards his PhD degree, 1993 to 1996. He has also received research grant awards from the National Research Foundation, NRF, in 2020, the Kenyatta University Vice Chancellor Research Grant 2013, and Lake Victoria Research VICRES Grant 2006, among other awards. He was nominated for a 3-month Seeding Labs Novartis Institute of Biomedical Research (NBR) in 2010 for a training and research fellowship program at Cambridge, USA from June to September 2010, upon which he received 2 LCMS equipment for research. Professor Nyambaka has extensive research in the use of analytical techniques in chemical analysis, focusing on nutrients in foods and environmental pollutants. In food nutrition, the emphasis is on processing procedures and their effects on nutritional and sensory values, bioavailability and bioaccessibility of nutrients and food formulations, while environmental pollution research focuses on levels of heavy metals and polyaromatic hydrocarbons, that's the PAHs, and methods of their removal and the impact of pesticide residues in the environment. He has authored and co-authored over 60 peer-reviewed research articles, proceedings, and book chapters, and presented over 30 conference papers in international and local conferences. He has supervised over 50 Master of Science and PhD students, and has currently a research group of about 20 postgraduate students. He has also examined several theses and referred several articles in reputable journals. He has a patent on food foundation developed from indigenous foods used for the management of HIV and AIDS patients. Welcome all the listeners this afternoon to Professor Nyambaka, who is very, very prepared to speak or share his what he has done so far. So welcome, Prof. You have 40 minutes. Thank you. Welcome. What could have happened?
Sorry, yes.
Yes, Prof.
Thank you so much for that elaborate welcome. I'm now sharing my screen, yes. Thank you so much for the invitation to this conference, and especially to give a talk on the work I'm involved in, basically on the aspect of promoting indigenous foods, the issues in the area of food and nutrition security. We will be able to share a bit of my thoughts and my part of what we have done in this kind of study. So thank you so much. Yeah, I am in the Department of Chemistry. This is at Kenyatta University, and I've been there as represented since 1988, being part of the faculty at the university, seeing it growing all through this time here. Bit of what I will cover, bit of some background, matching issues, results of some areas that we have worked in, just a few, not or just one or two areas, and a bit of a summary. Sorry, can go straight into my talk. Food security. It's a historical area that normally has been dealing with the overall regional, national, and the global food supply and shortfalls. The requirement of food. That was the original look at what food security could entail. But now it's going beyond just the food supply, because initially it was just the food supply, and if there's any shortfall. But now beyond the food supply, it includes elements of access, vulnerability, and sustainability. So it's an area that keeps on evolving, and even as we are right now, still there are some areas that are evolving or emerging up, so that we encompass the whole aspect of food security. As Kenya government is keen on reducing hunger and malnutrition, as emphasized in the Food Nutrition Security Plan of 2018, it is imperative that capturing the different dimensions of this evolving topic of food security, food nutrition, that it's captured all the areas, so that now the fight against food or malnutrition becomes important. So the evolution of food and nutrition security has been evolving right from the year before 2050, and it's getting in 1945, there was this aspect of food insufficiency in some areas, and therefore there was surplus from the developing countries that were giving to people who were suffering food shortages in some areas. So there was a food supply disposed from the developing countries areas that were in need. But by 1960s, it was realized that food aid actually hindered development in such areas. And therefore, the emphasis on food security now changed to food development, food for development. So that now the whole concept of security was looked at from that point. But in 1970, out of the crisis, the food crisis that was there, just before that, the donor countries now started realizing that food security is much more important, and assurance of that security is becoming the most important part of it. And at this time, some insurance systems were developed to assure food supply, and therefore the emphasis was given on food assurance. But by 1980, when the Green Revolution had happened, there was a lot of increased food availability was so much, and it was clear that the degradation of food is not due to the availability of food, but actually due to the individual groups, people able to purchase. So the concept of security now looked at a broad approach food security. What's the issue now became reducing malnutrition and hunger, and this was through the availability of adequate nutrition, not just availability of food, but adequate food and nutrition. So the emphasis now when hunger and nutrition. By the year 2000, now in the aspect of reducing food and malnutrition, it was now looked at as a process of overall development. As you are reducing hunger and malnutrition, it was seen that actually anchored in the overall development, reduction of poverty, and the achievement of the millennium development goal. So it's an area that has been emerging as we go on. Until now, we are able to have a definition of security that is fairly encompassing what the concept is now. Food security is achieved when it ensures that all people at all times, physical, social, economic, access sufficient, safe, and nutritious food, which meet their data needs, and the food prevalence for an active and so in the encompassing some of the areas that the preacher were not there when the FAO gave his definition. And this definition has four pillars, and the pillars start from the four ones: availability, access, utilization, and sustainability, where on the area of availability, the emphasis is on supply of sufficient quantity and quality, which depends on domestic production, stocks, imports, so that there is sufficient amount of quantity and quality. While access now looks at the aspect of individual and household adequacy of the food, where you look at political, economic, social factors, stable distribution, affordability, infrastructure, all those areas that will increase and access. Utilization is where you think of when food is contains enough nutrients which can be utilized as a adequate location which can now depend on safety, quality, nutritional knowledge, proper preparation, and environment. There is also the issue of stability, which deals with the permanent availability or access to the resources. through maintenance of the other three pillars, so that there's adequate supply. So that gives us now a broader picture of what nutrition food is. But as we look at these four factors, they actually we can group them into these four factors. The first one is when we look at the physical disaster, the aspect that will enable the accessibility of food, the availability of food. And this starts from food availability. Then you come to access of the food, and then you the evaluation of the food, which now concerns the nutritional status of an individual. But we also have to which affects all the three parameters in terms of the stability of all these other areas, so that the nutritional status can be affected. And therefore, we are now just having a concept of food security, the new shift of food and nutrition security than just talking food security. So the new concept and nutrition security will be achieved adequate food in terms of quantity, quality, safety, and socio-cultural accessibility is available and accessible for and satisfactory users and utilized by all individuals at all times to live a healthy and active life. A much more and you start looking at this in terms of how is the issue of food security growing up in the areas and the emerging at all times in our lives. So that now food security will be access to food, more daily access to food. One need to have a productive life. But nutrition security goes beyond and looks at securing that access, coupled with sanitary environment, adequate health services, knowledgeable care, foster good nutritional status through the life cycle across generations. So that now we look at nutrition security as very, very important when we talk about food security. We can be able to see nutrition security as an aspect that brings in or came in as we are looking at the malnutrition, the aspect of malnutrition. And the conceptual framework, which is used by UNICEF, trying to look at the aspect of nutrition security through a biological approach, where it is focusing mainly on nutritional status. Where food security basically focuses on economic approach, where it's food as a commodity, and actually that was the original concept of food security. We are looking at food as a commodity that make it available, and therefore secure food secure. But now, this conceptual framework is indicating the manifestation of malnutrition and what are the underlying causes. What are the causes that come here? So in malnutrition, we have immediate causes that basically are in the level of an individual. These causes, you look at them at the level of an individual, where inadequate diet would have affected the health of that individual. will be responsible for malnutrition. While at the same time, the health issue of lack of rare diseases or anything will result in malnutrition if it's availability of that food. And these two factors which are immediate causes, the health of a person who is taking that food is crucial for the person to receive or have the micronutrients for intake and agriculture. So we are saying here that these immediate causes of malnutrition will have underlying causes that bring up these immediate causes like inadequate access to food, because therefore there's inadequate intake, inadequate care for children and women and most vulnerable groups in society, and the insufficient health services and health environment. Because people are living in an health environment, become sick, and therefore they are consuming food, they are not able to absorb the nutrients in the body because of the sickness. And all these are the basic causes that are coming. Basic causes dealing with resources and control, all coming from the political factors, economic and so on. So they have this basic or underlying basic causes that will affect the underlying causes, which have an influence on the immediate causes causing malnutrition. So that as we understand the basic factors, then the fight against food and nutrition security can be addressed well. So what are some of the causes of malnutrition we can be able to look at in terms of when we look at an individual or in society? Major causes in terms of lack of access to health food, these are picked from the immediate causes of absence of micronutrients. Yeah, people can be taking a lot of micronutrients and that affects the health of an individual. A number of micronutrients are very crucial in body development, body maintenance, zinc, iron, potassium. We are talking of many other micronutrients, and the vitamins, the coming and some of them. Lack of water and sanitation, maybe reduce the issues. cut quite a number. We are talking about the first one. Direct relationship with the purchasing power, which is number 8, which has a direct relationship with hunger, nutrition, and food security section, as is true, and the rest. So this third relationship, we can be SDG 2, it will have an impact with a number of these SDGs, and therefore achieving what we are looking at. So my contention there is that as we look at nutritious foods, as we talk of availability of food that's nutritious and all the time, but the idea of bioavailability of this food is very crucial. Bioavailability. What is bioavailability? We are talking of it as a post-absorption assessment of how much of a nutrient has been absorbed in the body to be functional. That's why we are saying that you can talk of having a nutritious food, but until you are able to be sure of the bioavailability of this, then still we are a bit far off. And this is an area that is now becoming more of being understood, an emerging area in terms of food and nutrition security. The factors affecting bioavailability, of course, we will have either enhancers or inhibitors, which are in food that either can enhance, which is a positive aspect of it, but quite a number of food would contain inhibitors. But we also have issues with the digestion. And this comes in when we are dealing with, in terms of, is the person sick? How is the health of that person? The health of that person will determine the digestibility of the food and therefore the bioavailability of the nutrients that we need to have. Then the third cause or effect, factor that affects is the individual variability. Male, female, children, and all these factors will determine how much of the nutrients from food is bioavailable. It's bioavailable. And therefore, we need really to go into this area as one way of contributing to the nutritional security in this country. An example is when some studies was done, I think in Busia, where a community that was consuming cassava because of the major, Busia, I think the coast, one of the two regions where cassava was introduced, assuming that it has high levels of these nutrients, and therefore by consumption of this, it was expected that effects like stunting and so on would be reduced, but there was no effect when that was made available, indicating that the bioavailability of the nutrients from this plant food is not necessarily the same as the levels that we know they have. And therefore, there are some strategies of dealing with bioavailability. One of the areas that was understood much earlier in terms of bioavailability, iron, availability of iron from different foods. And therefore, there have been various ways of improving bioavailability. But normally this has targeted on fortified foods than just the natural food from the plant foods or the animal foods that we know them. So one way they have done is to, of course, fortify the food, making sure that the food is in a place where it's bioavailable, using iron salt, or even element of iron, or even micro encapsulation, so that that would be able to have the element bioavailable. The encapsulation or the reduction of the iron particles makes it easier to be absorbed into the body. And that's the concept that now is being developed. But we can also be able to look at the food matrix. You add in a food matrix food in, ion enhancers, like ascorbic acid, or even adding the animal tissue to do that, but that needs to be understood much more. And also looking at the food matrix, whether it contains the ion inhibitors, like the phytates, phenol, polyphenols, calcium acts against the presence of iron, and so on. So this aspect of what is the food matrix made of? Is it likely to enhance or to inhibit? And of course, you target the population, as you do your fortified foods, pregnant women, the women at childbearing. We look at these emerging issues of bioavailability and vis-a-vis the processing or cooking of these foods and so on. So we'll try to look at quite a number of intervention studies where we look at special groups like children or people living with HIV and AIDS, so that we are able to see if intervening with the food with high levels of this nutrient, is there an impact, is there a change? We have also been looking at food processing and their impact on the levels of micronutrients. And beyond just looking at the level of micronutrients, we have also been looking at the bioavailability of these micronutrients with processing and with the different food matrices. So the first one is where we looked at the intervention studies on people living with HIV and AIDS as a project sponsored by the Victoria Basin Research, the VICRES. And the focus of research was basically to use indigenous foods and check on air utilization, check on the micronutrients analysis, the levels, and of course find out what effect it will have on people living with HIV and AIDS in the process. Of course, as we were looking at, we worked with this project in Western Kenya, in Busia, where we worked with some young people that were there. And we were looking at why are people not taking indigenous foods? It became out very clear that people are shunning out of indigenous foods and they're going out to the quick foods that you can get, the mandasis and these other foods that are made from just flour. And we realized that once the of course, the indigenous foods contain rich in nutrients, contain high levels of the micronutrients. They are actually to turn a corner, and people are encouraging the refined foods. So we decided just to work with these people living with HIV and AIDS, and see how this will have impact on them. Why were we interested in nutrition and HIV, AIDS and HIV disease? Because the fact that nutrition, poor nutrition, will make the patients not to be able to fight with the disease and end up infectious diseases. And this ability not to fight the diseases will increase vulnerability to more diseases other diseases coming in, then it leads to poor health, and then progression to the stage of age. But increased nutritional needs, and of course this one also leads to increased nutritional needs. People not taking food very well, reduced food intake because of the elevated numbers, and this also led to increased loss of nutrients, and therefore goes back to the effect of nutrition. So the HIV has impact on all these areas, and therefore we thought of why can't we come in and see what we can be able to help. So the role of food was to manage because of the patients actually require 10 to 50% more of the calories when they are in that stage, and 50 to 100% protein per day than the usual, the one who doesn't have. And we also know that the vitamins and minerals, the microelements, are very important in terms of mitigating the transmission and propagation. Some of the elements we looked at, zinc, selenium, vitamin A, E, and C, which has shown to actually have an impact, reduce symptoms, illnesses, and so on. And therefore, we were dealing with the indigenous foods. We processed them, and through drying and cooking, looked at the effect of that. Then, of course, we made formulations, which we were knowing that they contain high levels of these micronutrients. And we looked at the vulnerable groups so that we had both intervention groups and the control groups in those kind of studies. And some of the statistics, the results we got from here are that these indigenous foods are not accessible. Quite a number of people are saying that's 49, 94% of the people say they are not accessible. And we went to find out why they are not accessible. Quite a number of people are saying they are not affordable, which means they are depending on buying of these foods much of the time. A few are not saying they are not available, others they don't know why they were not consuming them. And therefore that came out very clear, and we were able to start thinking of how to now talk to the people so that they can be able to think in terms of that. And in terms of the major group, the feeding habits, a majority of the people were eating the cereals, that's ugali, about 90%. So the meal was basically ugali 90% coming from cereals. And the accompaniment was basically vegetables, but mainly cowpea leaves. Mainly cowpea leaves was the majority. But we realized that animal food was very low, 19%. Quite very few were affording to have any kind of animal. In their original forms, and we realized quite a number of these vegetables that are found in that area, but are not available to the people. Very, very common in the area, Arsenal area, but not very common Arube area. And secondly, we were able to have another friend of groups, children in arid and semi-arid regions, and this one was in Machakos. We went to Machakos and we had an intervention study where we were looking at basically the vitamin A deficiency in the preschool children. And this is why we were looking at it, is because those areas, they lack, there's a big lack of food, and much more so the indigenous vegetables, because the place is wet in a very short time of the year. Most part of it is dry, and there the vegetables can be found within a very short time, unless those who are staying in towns or marketplaces where they can receive, but those who are received through buying, but those who are staying in rural areas, it was a challenge. The children were basically eating maize, boiled maize alone without anything. And therefore, we decided to look at promoting these vegetables in schools as as school gardens, and therefore coming up with even ways of preservation of these foods after that. And then we realized that quite a number of these vegetables here are in terms of which ones are more commonly used. Something like spinach was common there. Cowpea leaves, I think because of you can be able to stay for a long time, was coming in as the most common. Method of preservation can do drying or even sun drying. Drying that is in the shade and sun drying. So the preferences of these foods now comes, we were checking on the preferences of these vegetables, like do they like them when they are fresh or even when they are sun-dried? The majority, of course, is when it's fresh, except just a few would take nightshade. But the common vegetables were, of course, cowpea leaves, spinach, and pumpkin leaves are the most common in that area. And these are just the levels of health in terms of processing. We were wanted to see if in the three different vegetables, like cowpea leaves, amaranthus, and cabbage, differing in terms of the fiber content, what is the effect of processing? That's basically it was fresh, cooked, sort of dried, and sun-dried. And we realized that the processing, cooking reduces up to about 77%, 76% for amaranthus, and cabbage, it was 95%, but the solar drying and the sun drying reduced further to around 60 or to just around 57 to 62. So availability studies is basically going on. It's as part of our work we are doing. And I didn't have results. I didn't put the results here, but it's an area that is going on. Some work, of course, we published on bioavailability in this area, dealing with microelements in vegetables, cereals, and mushrooms, and also dealing with the vitamins in raw processed vegetables, cereals, and mushrooms. But another area that we are also trying to think more in bioavailability, apart from processing, what about the chemical form of these nutrients in food? How much does it affect the bioavailability? And this bioavailability, we have been doing it in two forms. One is after the intervention, we are able to find out the differences in level of the nutrients in blood. That's after intervention. So we check the levels before and after intervention. And we realized actually, it's only that I didn't have my results here, there's a significant increase. So there's bioavailability when these vegetables are consumed, especially came out very well in the children in the arid and sub-arid areas where earlier on they were not consuming these vegetables, but when we introduced the school garden, grew the vegetable in school and the children were consuming them as part of their lunch once a day, there was a significant increase in terms of the bioavailable elements. That came out very clearly. And we are also able to do the analysis based on the in vitro approach, the in vivo on in vitro. The first one was in vivo, but we can do an in vitro approach where we simulate the processes in the stomach such a way that we can be able to measure the bioaccessible nutrients, and that will tell us a bit more of the bioavailability of these nutrients. So it's an area that's going on, but we are expanding it to include the chemical form. Therefore, in summary, food and nutrition security is crucial in the achievement of acidity in Kenya and in the developing countries, where indigenous foods are being promoted. Most of the time, we need to promote them and processed in a way that micronutrients are readily bioavailable. By understanding the essence of bioavailability of micronutrients and its enhancement with the desire for nutritional security to be achieved. Yes, we are in this together, and let us soldier on. Thank you for this time. Thank you.
That talk. And I believe those who are tuned in, you've been educated on, the indigenous vegetables. And now, after having taken us through on indigenous foods, he has elaborately explained the meanings of the terms used: food security versus food supply, accessibility, vulnerability, sustainability, and all that. And even with the most current look at what it all means when we talk of food insecurity, and looked at even later on the current research areas on intervention with special groups. So I believe, ladies and gentlemen, we've been educated enough, and definitely you may be having a question to put to Professor Hudson Nyambaka, and I want to open up to the Professor Luther. You can ask Professor Nyambaka.
Okay, thank you, Professor Sweet. Thank you, Professor Nyambaka, for the enlightened talk. I think you have made us appreciate chemistry, that you can actually extend it further and use it to solve the problems affecting the society. Because this area of food security, food nutrition is key. And we know that it's even one of the big four agenda of the national government. I just have a comment. I saw your talk. At least you have discussed about food security or food nutrition and health. But there's a component which I was waiting for, but I was not able to see it, because we know Kenya as a country is known for sports. I thought maybe we could also comment on that, food security and sports, and food nutrition and sport, how they are related and how they can be improved. And then the other thing that I might want you to comment on is about the GMOs. I think that one can also come in as a factor when you're talking about the nutritional level. I may not know about the bioavailability, but maybe you can comment on that one. And then lastly, I believe as a country, we have a lot of food, and most of them occur naturally, but what we have done is we have neglected what occurs naturally, the indigenous food, and we have gone for so that we can focus on what is available locally, which is healthy, free of chemicals, and the safety for consumption. Thank you, Professor.
Professor Nyambaka, you gotten the question. Maybe let's take another. He had 3 questions. Okay, let's have a response, and then we get the other questions. Welcome again. You gotten the three questions from.
I think I got city 2, one on sports, GMO, and the other one was?
The other one was on how can we change the mindset of our people so that we consume what is locally available, which we know is free from chemicals and safe and very healthy, so that we don't have people depending mostly on food stuff imported from outside, which some people feel they have maybe some high quality in terms of nutrition, which may not necessarily be true.
Yeah, thank you. Thank you, Professor. Yeah, these are really pertinent issues in terms of nutrition, food and nutrition security. As we find this aspect of insecurity that cuts across or that would have an impact in the general development of an individual, of people, and so on. Yeah, the first one, the nutrition and the sports. Definitely, I think I mentioned somewhere very, very briefly as I was passing it through, that nutrition and sports is very, very crucial. That the people of sports have to live a disciplined life in terms of the nutrients they are taking in, and so that the body can be able to have or to be accessible to all the nutrients that the body requires, and therefore you remain fit for your sport. So definitely there's a direct relationship, so only that studies have not been, not many studies have come across dealing with relating to the type of foods that the people of sports take with their nutritional status. That has not come out very well, but I know there's quite a number of literature. And this is because mainly the people of sports are normally in a specific regiment. Their food intake is mainly like sort of control or something. And therefore, the understanding is that they're in the point of getting the enough nutrients they need in the body. So they are nutritionally secure, we can talk of. But of course, that's something which can be understood even much more in terms of how do you relate the nutritional security and sports in terms of the consumption of the locally available foods the way you are putting down here. So that's an area that can be expanded on. The area of GMOs, yeah, there's this controversy of GMOs not being accepted. They are being accepted, they're being consumed. These are part of what we can talk of as exotic foods. They are being, the local foods are being modified so that they can be able to have either high levels or they can be able to produce more in terms of having the accessibility of the food. But the question now remains is, how bioavailable are the nutrients from these GMO foods? That becomes the question, because the GMO is dealing with the provision, the quantity, the access to quantities of food, making enough food being available. But are we addressing the issue of bioavailability, bioaccessibility of these nutrients that becomes an issue that can be looked at as we promote GMOs. Are we sure that the intention is supposed to have is achieved in terms of nutritional accessibility? And then the third one, you're talking of, you're asking whether, what we can be able to do to enable people, make people go for the locally available foods than going for the exotic or those ones they feel like contains more nutrients. It's a matter of cultural, socio-cultural issues that needs to be addressed. And when one looks at the Kenya Food and Nutrition Security Plan for 2018-2022, they have a section dealing with that in terms of helping people understand that the local foods are actually so nutritious than the food you think you're buying from out. You go for the local vegetable at home, the mlenda, for example, or for the manago, for example. They contain these high levels, but now what the question we are also asking ourselves is, the high levels are there, yes, but are they bioavailable? That's what we are addressing right now to deal with the issue of you are consuming this thinking that it's giving you the enough nutrients, but you may not be giving because of the food matrix under which the nutrients are in. So the promotion of the locally available vegetables will deal with issues of socioeconomic, sociocultural, socioeconomic issues. And that's why when we were going into these two area studies of intervention, the idea was actually to promote these vegetables. And eventually people receive them, but the issue comes in, are they able to continue now growing them on their own? And thereby having these vegetables around, foods are available or not. You know, that's the aspect. We have gone too much in economic terms, in terms of things that will bring us money, not things that will give us more food or food that is nutritious. Thank you.
Yes, you can ask.
Thank you, Chair, and thank you, Professor Adson, for your presentation. I just have one question. What could be the angle or the contribution of palatability to the impact of food preferences and probably causing some of the deficiencies in micronutrients in the country, especially to the young generation that probably they are not used to some of the indigenous foods that we have? Thank you.
Yes, Professor, let's have another or two more questions. I hope you gotten that question from Dr. Tway. I see there is Kemei. Mr. Kemei, is it? Yes, you can ask your question. And she still needs to ask. She has another question.
Okay.
I see a hand from Dr. Tway again. Dr. Tway.
Sorry, Chair. I need to raise it down.
Hello? What? You've already raised it. Oh,
Yes,
maybe.
Chair, sorry. I don't have any other question. I don't have any.
I see no other question. I'm not seeing. Okay, Dr.
Thank you, Professor Nyambaka, for that wonderful presentation. So my question is just simple. I wanted to find out the difference between solar dried foods and sun dried foods.
So another question, those are two. Maybe we allow Professor Nyambaka to answer those.
Thank you, Chair. Let me start with a question from Dr. Tway, Vivian. Thank you for that concern, because it's a real concern that we have in terms of the young generation actually not only shunning off the indigenous vegetable, but most of the foods that are taken by the older people, they think that they don't need to be in that kind of food area. Yeah, it's a concern that I don't know whether it's dealing with the palatability of the food, but more of the preferences. The concept of feeling like the packed and processed foods are quick to prepare, are palatable, yes, you say, but yet they are not interested to find out the relationship with those palatable foods, that processed foods, and aspect of nutrient, whether they contain nutrient or not. So that now the market is going to prepare foods that are palatable so much, but ignoring the essence of nutrition. So actually they have gone back to the aspect of providing food, which is processed and very fast enough that the young generation will quickly rush to, without thinking in terms of the nutritional aspect of it. Remember, this is a journey moving from just the access to food to the access to food that has nutrients. So it's like they're going back actually, and it's a concern for, we should be able to be concerned in this area and deal with it in the aspect of how do we handle that kind of an area. So, but the aspect of actually, I know you may talk of the indigenous foods, like you talk of the vegetables, indigenous vegetables. They may not be acceptable to many people. One is because some vegetables are cultural in nature, yet they contain high levels of these nutrients. You get one culture is not able to consume these vegetables because they don't think it's a vegetable at all, while another culture is taking it. So that aspect of moving out of the cultural boundaries, the cultural influences, so it needs to be addressed. I know like we are doing a project on the use of insects as food. Quite a number of cultures don't appreciate that, but there is a place where the consumption of insects is a food that is required so much. So culture is affecting the consumption of the indigenous food, some of them which are very good, but also the aspect of their being available, these foods being available is an issue that needs to be addressed. As much as we want to treat our people, to take our people to be consuming these foods, are they going to be available? Indigenous vegetables, sometimes they are not freely available. It's only the people, some people who can be able to go for them and so on. So it's an area actually that we need to address. Secondly, the second question by Joshua, the difference between solar and sun dried. Solar drying is when you are drying a vegetable using the sun, the solar, but not directly, under shade. Quite a number, if you go some old generation, they used to dry vegetables not directly in the sun, but they put under the shade of a tree, but eventually the vegetables dry. And these people knew even before then that if you dry directly on the sun, you would affect the nutrients in the food. You just kill them, all of them. So, but solar drying maintains the nutrient, but it removes the water molecules. While sun drying is a direct drying using the sun. Like we dry the maize you put them in direct contact with the sun and so on. So that's the difference between the two. Thank you.
Thank you very much, Professor Nambaka, for responding to those questions. And I want to say thank you so much for participating in this conference. And just because of time, maybe we shall stop there with you. But I would request you that you can still be tuned in and continue possibly to hear what is going on after your presentation. Otherwise, I want to say thank you so much, and we appreciate your participation, and we look forward to much more connections and links with you, and God bless you. So I think that's all.
Thank you so much.
We appreciate. I think is that so, Chair, I think I'm through. Maybe now we get to the next the parallel sessions, or those of us who have not had lunch, who can be told what to do next. Thank you. So,
Recording stopped. Recording in progress.
Varying over a given time interval. So the length of growth does not immediately change until the amount of growth mitosis factor reaches a critical value, which is noticed after a given period of time. So when the amount of R increases with time, we have the length of growth being also increasing. So the effect caused by the growth factor causes the length of growth to also change. Now, in conclusion, the solute concentration taken as fertilizer concentration in soil media over a given time increases, causing an increase in growth mitosis factor. And finally, this results in increase in apical, that is the vertical length of the plant. Such scientific findings play a big role in explaining processes that can improve agricultural practices, especially the use of both organic and inorganic fertilizers. Through mathematical modeling, we can be able to simulate and also explain agricultural processes by formulating and solving equations of fluid flow and equations of growth. Now, my recommendation is from the research, the concentration of fertilizer and growth mitosis factor coupled with the length of growth of plant were emphasized, leaving other factors out. So in future research, the velocity of flow of fertilizer solution through soil medium can also be modeled and studied to see the effect of the speed of flow of the mixture, that is the fertilizer mixture, on plant growth. Again, further research can be carried out to check how a specific fertilizer concentration in a given type of soil can also affect plant or growth. Thank you for listening to me. That is the end of my presentation.
Thank you, Timothy. Elaborate. So
Hello.
Yes, can you hear me, Timothy?
Yes, yeah, I can get you.
So thank you so much for the good presentation.
Yes.
I open the floor to any reaction, comments, or question. Members, anyone with a question to Timothy or a comment?
He needs to work on the slides. Yeah, otherwise it was a good presentation.
All right, thank you for that. So I think we can just proceed to the next presentation. Once again, Timothy, thank you.
Okay, thank you for listening to me.
So I don't know if we have Dr. Jenson Murithi. Dr. Jenson Murithi, are you with us? If not, John Buru, are you with us? I must give us a minute to inquire on the abouts.
Brown bottles. The samples were stored at 4 degrees Celsius. in cool boxes, and extraction was done within 48 hours. For the samples that were not extracted within 48 hours, they were acidified to a pH of between 3 and 5 using formic acid. Then the samples were pre-treated before extraction, and in the pre-treatment stage, the samples were filtered. They were then compressed using 0.25 grams of sodium EDTA, and the pH was then adjusted to 7. After that, the samples were extracted using solid phase extraction cartridges, and the samples that were extracted were actually at 250 mils. After extraction, elution then followed, whereby the samples were eluted using methanol. And after elution, the samples were reconstituted to 1 mil using reconstituting solution, which is made up of methanol, HPLC grade water, sodium EDTA, and formic acid. After reconstitution, the samples were then analyzed using ultra HPLC interfaced with high resolution MS, then data analysis followed. On that slide, we have results of the APIs that were detected. 50 APIs were analyzed, and out of the 50 that were analyzed, 16 of them were detected in the samples that were collected along the river basin. And out of the 16 APIs that were detected, they were categorized into 5 categories. We have the analgesics, antibiotics, antiretrovirals, psychiatrics, and beta blockers. We had 2 analgesics that were detected: ketoprofen and paracetamol, chlorothromycin, metronidazole, nalidixic acid, sulfadoxine, sulfamethoxazine, and sulfamethoxazole, and trimethoprim were the antibiotics that were detected. Then we have 3 antiretrovirals, 2 psychiatrics, and 2 beta blockers. Now, when you look at that graph, you'll find that sulfamethoxazole was detected in all the 24 sites. We had 22 sites along the river and 2 wastewater treatment sites. So in all the 24 sites, sulfamethoxazole was detected. And of course, someone might wonder why sulfamethoxazole was also detected in the upstream sites. The upstream sites constitute of the 5 rivers upstream, which were shown in the map. So you find that sulfamethoxazole was detected upstream, mainly because this drug, sulfamethoxazole, is mainly used for veterinary use. And when you look at the upstream regions of the river basin, the upstream region constitutes of the Kiambu region, the entire Kiambu region, where we have the source of the river. So there's a lot of small scale, or we have small scale farming, and they use sulfamethoxazole for veterinary purposes. The second compound that had the highest detection, or the second highest detection frequency, was seen for carbamazepine. Carbamazepine is a psychiatric drug, and this drug was mainly detected in the midstream regions, the regions within Nairobi area, in the areas that are adjacent to the informal settlements, that's the Mathare River, Ngong River, and Nairobi River. Next. On this slide, we have a heat map showing level of contamination. Now,
Faith, you have 2 more minutes, so please summarize.
Okay. So when you look at this heat map, when you look at the areas that are shaded green, those are the upstream regions. And you find that the levels that were reported were between 0 and 54,000 nanograms per liter. And when you look at the heat map again, you'll realize that analgesics had the highest concentration, total concentration, in the midstream regions, where we have Mathare River, Nairobi, and Ngong, which pass through the informal settlement areas. Next. Then we have APIs concentration along the river profile, and the two wastewater treatment plants. So when you look at this box plot, you'll realize that the area or the site that had the highest concentration is Ngong River before Nairobi Dam. That is where the river passes through Kibera slums. That is where we had the highest concentration of APIs, and the API that had the highest concentration that was reported was paracetamol, followed by lamivudine, which is an antiretroviral. Next. Then APIs, total concentration in all the sites. Sulfamethoxazole, as I mentioned, had 100% detection, and that is why we had very high concentrations along the river. Zidovudine, an antiretroviral, had the lowest total concentration, followed by nalidixic acid, metoprolol, and ketoprofen. And generally, you can see antibiotics had very high concentrations, which poses a serious health risk. Next. So in conclusion, the sources of APIs along the river were found to be from uncontrolled release of untreated wastewater from informal settlements, discharge from industrial or industries where drugs are formulated. We also have landfills, that's from Dandora, which is the biggest. We also have veterinary medicine from upstream sites and ineffective wastewater treatment methods that are being used. We find that in most, or actually the two wastewater treatments, APIs were detected. Water quality along the Athi River basin, you can say that the water is contaminated with APIs, and therefore there's potential risk to humans and to livestock. And the potential risk that is posed by these APIs is that, one, we have drug resistance, especially antibiotics, and of course, effects on aquatic ecosystem. Recommendations: there is need to come up with point of use water treatment methods, especially downstream where the water is being used for domestic use, so that these APIs can be removed before the water is used. Then it's important also to look at assessment of APIs in food products from irrigation, because irrigation is done downstream and also within the midstream areas. So there's need to assess APIs in food products, and also assessment of APIs in milk from cattle along the rivers, more so the antiretrovirals. Next. Acknowledgement: this research was supported by BLIR US, the South Initiative, Belgium, Ghent University, University of Eldoret, and Water Resources Authority. Thank you.
Okay, thank you, Faith, for that good presentation. I'll now open the floor to members to make comments and questions. I will give 3 minutes for that to make up for the time lost. So for the other two presenters, make sure you stick to the 15 minutes. So the floor is yours if you have any questions. If you cannot unmute yourself, please put your questions or comments in the chat and I will read them. I guess as members are thinking of questions, maybe I can ask one. Now, a lot of these antibiotics and the drugs that you've looked at are drugs that we take. So the question I'd like to ask are the drugs that you detect in the water the same formulation as that is given, or are they metabolites, like once it goes through the liver, is it metabolized into a different structure, or is it the exact same? It's excreted the same way into the water as what we take in? Thank you.
Okay, thank you, Doc. Most of the drugs are excreted in the same way, but some are degraded in the body. And a good example is sulfamethoxazole. Sulfamethoxazole is degraded into metabolites, which when they are detected, the detection of these metabolites give the sum of the sulfamethoxazole that is in the samples.
Okay, thank you. Are there any other questions from the floor? I can see Dr. Makwali, please go ahead. Then after that, Professor Luta. Dr. Makwali? Dr. Makwali, we can't hear you. Please.
Thank you, Chair. Can you hear me now?
Yes, please
Hello?
proceed. I can hear you now. Please proceed.
Okay, thank you. Mine is just to congratulate the student on the work that she's doing. It's a wake-up call for all of us. I think this is a study that needs to generate a bigger conversation. As the science world, we need to find out what is really the way forward, because it is a bit threatening that all these active pharmaceutical ingredients are really going to pollute our water masses. So what would this mean to us in the near future? That is a discussion maybe we need to have, and we need to think, what are we going to do so that we can stop or eradicate this contamination that is going on? Thank you.
Thank you, Dr. Makwali. Professor Luta, please go ahead.
Okay, thank you, Dr. Bisem. Thank you, Faith, for the presentation. It's very interesting. I think Dr. Makwali captured what I wanted to say. I just wanted to comment that actually this is an area of interest, knowing very well that at the moment we don't have clear policies when it comes to disposal of pharmaceuticals. And nowadays, these clinics are, dispensaries are just set everywhere. So I think we need to look at this work with the seriousness that it deserves, and it will go a long way in solving the problems affecting environmental pollution. I think I just wanted maybe Faith to comment on two things. One is, did she do seasonal variations of the pollution of these pharmaceuticals? And then maybe quickly, you can just mention what are the effects of some of these pharmaceuticals, because the fact that they're just being there in high concentration may not tell us much. Are there effects, toxic effects of these pharmaceuticals which have been documented and are known? Thank you.
Okay, thank you, Dr. Judith, for your contribution. Thank you, Professor Luta, for your questions. The first one was on seasonal variation. Yes, this is work that is ongoing. This is work in progress. These are results for the first sampling campaign. We did 3 sampling campaigns. So these are results for the first sampling campaign. Data for the second sampling campaign is out, but we're still processing the data, and also data for the third sampling campaign. So in due course, I'll give comprehensive results in the near future on seasonal variation. Secondly, on effects of pharmaceuticals. Yes, these pharmaceuticals pose a serious health risks. As I mentioned, the first risk, which is a major risk, is about antibiotics resistance. Earlier on in the mornings, our keynote speaker mentioned something about antibiotics and the need to do continuous research in and the use of antibiotics and development of new antibiotic drugs because of resistance. So this is one of the reasons why it is important to come up with policies to check on release of antibiotics into water bodies. There is need also to check on technological methods that can be used to remove these APIs from water. And this is an area of concern. And in my second part of my research is ways of coming up with methodologies of removing these APIs from water, which again, maybe in due course, I'll also give my input. Thank you.
Thank you, Faith. We are still trying to connect to the other 2 presenters. So maybe I can take maybe one more question as we attempt to do that. Dr. Macaulay, do you still have a question? I can see your hand is raised.
Sorry, Chair, I should have lowered it.
Oh, okay.
But just to make, I'm just thinking also, could there be also underground seepage to other water masses? Is there a possibility of underground seepage from the source of contamination to other areas that might not be contaminated?
Yes, Doc, there is data to that effect. A research that was done by Correge et al. In 2016 showed contamination of groundwater. And I said groundwater is water drawn from the wells, the shallow wells. And in that study, it showed that water from the wells tend to be contaminated with APIs, mainly due to seepage and percolation into the groundwater. And again, remember that in most of the rural areas, most people use pit latrines. So you can imagine when someone takes antibiotics or these APIs or these drugs, once the waste are in the pit latrines, you can guess what happens. Whenever there is high rainfall, of course, we expect that I will have a seepage into groundwater, or groundwater will get contaminated. So there is data to that effect. And again, I also want to mention that there is no regulation, as Professor Luther mentioned, there's no regulation on the disposal of these APIs. And secondly, there is no data on the maximum allowable limits as for the other contaminants like heavy metals, nitrates, phosphates, fluorides, and so forth. So for APIs, this is an area that really needs a lot of work to be done so that some regulation is done, and also the maximum allowable limits be set so that at least we are able to know what levels are safe and what levels are beyond the limit. Thank you.
Sorry, our presenter happened to join the wrong room, so we're just rescheduling. We're asking her to join this one. So just bear with us for a few minutes. I'll just say thank you, Faith, for your presentation. This is some really good work, and I hope that you will continue it, because this is clearly showing a serious problem that we are yet to properly deal with and figure out what are the possible solutions. So I'll give us maybe 1 minute to just wait as we get her to join, and then she'll present from 4:02, hopefully. Thank you. The rapporteur, my rapporteur to put up the presentation. So give us 1 minute for that. So our final presenter for today is Ms. Miriam Wepukulu. She is a PhD student at the University of Nairobi. Miriam, the floor is yours. Proceed.
Okay, I'm Miriam Natecha Wepukulu, a PhD student at the University of Nairobi. My supervisors are Dr. Peter Wachira, Professor John Nderitu, and Dr. Paul Sifuna. The topic is optimization of conditions for biodegradation of chlorpyrifos in selected Kenyan dairy farms. The next slide.
Continue, please.
Okay.
And when you want the next slide, just say next so that they can press the next slide. Thank you.
Okay. So annually, there are in most parts of Africa, there are deaths of cattle caused by ticks, and they are prevented by highly poisonous diverse compounds known as organophosphates, which are the common ones used in Kenya include chamaphos, parathion, malathion, chlorpyrifos, among others. The chemicals contaminate soil, water, and 99% of them cause damage to beneficial biota. Next. Biotic and abiotic pathways can degrade the pesticides in soil. In some pesticide-contaminated environments, autochthonous bacteria have developed over time termination. Next. In the statement of the problem and justification, livestock farmers continuously use and dispose acaricides, leading to contamination of soil, air, and groundwater. This leads to acute illnesses like cancer among humans, killing beneficial biota in these environments, habitat destruction, and failure of reproduction of birds, as well as forest reduction. So there is need for an environmentally friendly method to address these residual effects of OPs and optimization of conditions for best biodegradation. Next. In the hypothesis, environmental conditions do not have any influence on biodegradation, and the objective was to determine optimum growth conditions for OP biodegradation, including temperature, pH, and OP concentration itself. Next. The contamination of acaricide polluted soil in dairy farms is one of the significance of the study, then provision of low-cost method of degrading pesticides. Next. Experiments were done in Mount Kenya University Laboratory on optimizing pH, temperature, and CP concentration. Next. Samples were collected from Nakuru, Subukia area, where there was soil sampling by use of soil auger, bacterial isolation from samples using enrichment method in MSM medium, then characterization of bacterial isolates using Gram-stain, catalase, indole, urease, methyl red, citrate, among others. The grouping of the cultures was based on morphological and morphological characterization. Then there was molecular characterization of the isolates. Next. There was genomic DNA extraction, then PCR, polymerase chain reaction, gel electrophoresis, as well as 16S ribosomal RNA gene region sequencing and gene blasting in the molecular characterization. Next. Those were the the biochemical characteristics of the strains, whereby for gram stain, all of them were gram negative for catalase. Most of them were positive oxidase. Most of them were negative, and the rest are as shown. Next. The genetic diversity of the isolates. The identity of the revived study isolate was confirmed by sequencing the amplified fragment of 16S ribosomal RNA gene region, which revealed that isolates belong to different bacterial species. There were 7 bacterial species to which they belong to according to the bands in the from MWA to MW1 to MW7. So the 8, there is no band because that was a control without any isolate. Next. Following the NCBI gene bank blasting of the 16S ribosomal RNA sequences of the bacterial isolates, all the isolates shown similarity of 90 to 100% in the gene bank with query coverage of 98%. The isolates were identified to belong to Pseudomonas, Alcaligenes, Bacillus, and Brevundimonas. Alignment and sequence comparison of 16S ribosomal RNA gene study sequences show that the study isolates were closely related to non-bacterial lineages. Next. That is the phylogenetic tree for the isolates, indicating the accession numbers and the close relatives. Next. So that is the table showing the isolates from MW1 to MW7, their accession numbers in the gene bank, MZ35982.2.1, all the way to MZ35982.8.1. Then the organisms with the most similar matches are as shown. That is Alcaligenes faecalis, Bacillus wenhenstenensis, Bacillus thuringiensis, Alcaligene species, Pseudomonas species, uncultured bacterial clone, and Brevundimonas diminuta. The accession numbers are as shown. The similarity was from 93 to 99, while query coverage was from 98 to 100. Next. For the determination of the optimal growth conditions, there was first of all chlorpyrifos preparation of chlorpyrifos from the stock. Then there was mineral salt medium preparation with all the ions. Then bacterial isolates that were previously isolated, tetrazolium chloride preparation, and incubation at 30 degrees Celsius. Next. All the 7 isolates change from colorless to purple after tetrazolium degradability. So in the medium that had tetrazolium without the isolates, especially the control, it remained colorless. But where we had the isolates were termed as able to degrade to degrade CP by change of tetrazolium from colorless to purple. So isolates degradation results are shown against the control, which was MSM and tetrazolium chloride without the isolate. That one did not change to purple. In all the experiments, a purple precipitate formazone was formed. So that was an indication that the isolates could be able to degrade CP.
Miriam, we have lost you. Miriam, are you there? Miriam, can you hear us?
at the corresponding pH. pH 7 was optimum, with OD ranging from, so all the isolates change from color, sorry. The next slide.
Miriam, we had
OD,
lost
highest
you.
OD at day 5, with the OD ranging from 0.6 to 0.85. Most isolates were neutrophiles at pH 7, except MW7, MW2, and MW3 that were alkalinophiles. Next slide. So for pH, pH, different pH at OD 480, most isolates were alkalinophiles still. Most of them were alkalinophiles and others were neutrophiles. Day 4 was optimum for CP degradation. Next. So growth of isolates at different temperature at OD 600 nanometers. So growth of isolates was best at 25 degrees Celsius for all isolates, except Ma2 with high OD 0.45 at 30 degrees Celsius. Next. However, the degradation temperature was 30 degrees Celsius, as much as most of the growth occurred at 25 degrees Celsius. So for degradation, the best temperature is 30 degrees Celsius for all the isolates, and it was significantly different from one isolate to another. Next. For the CP concentration, 150 ppm was the best CP for degradation of all the isolates except MA1, which degraded best at 100 ppm. Then there was no significant difference between 150 ppm and 100 ppm for most of the isolates. So decomposition can occur best at 150 or 100 ppm. Next. Yeah, so that is for biodegradation. So there is no significant difference between 100 and 150 in most of the isolates, as much as 150 was the best for a few isolates, including MA7 and MA1. So for the rest, there was no significant difference between 150 ppm and 100 ppm. Next. So most of the isolates grow best at temperature 25 degrees Celsius, but for degradation, the temperature is 30 degrees Celsius. That is the optimum degradation temperature. Then most of them are neutrophiles and a few are alkalinophiles. An incubation period of 3 days is good, but best degradation time is 4 days. Then optimum CP concentration is 150 ppm, but not significantly different from 100 ppm for degradation. Next. So in conclusion, autoclonal soil bacteria have shown capability of degrading OP acaricides. pH, temperature, and CP concentration influence the degradation and it increases with time. Next. So in the recommendation, molecular techniques should be employed to confirm the presence of bacteria from other samples, including water and milk contaminated with organophosphates. Then also soil should be checked for pH prior to application of OP for ease of biodegradability determination of conditions. An acknowledgement goes to my supervisors for their tireless support during the project. Next. Thank you. That is the end of my presentation. Any questions or concerns are welcome.
Thank you very much, Miriam. Thank you for presenting within the time frame of 15 minutes, even though we had some disruptions, and I also apologize for those disruptions. So I open the floor to members for about 3 minutes where we can have any comments or questions from the floor. If you cannot, if where you are is very noisy or you cannot speak, please put it in the chat. So kindly raise your hand so that I may choose you to make a comment or to give a question. Maybe I can start, Miriam.
Yeah.
Your research is in concerning organophosphates, which are used to treat ticks.
Yeah.
And you use chlorophos as the representative of organophosphates.
Yeah.
So maybe I can just ask why you chose that, and is it a type of organophosphate used as a pesticide? Is it a different kind of compound? And if so, why did you choose that compound? Thank you.
Okay, we did some survey in different study areas, and for Nakuru, it was seen that chlorpyrifos was the widely used. So most of the farmers used diazinon. So diazinon is a compound that contains chlorpyrifos. So that is what made me to narrow down to diazinon and the chlorpyrifos that is contained in it. Yeah. So like I was looking at these compounds that are used to treat ticks. From the survey, diazinon was mostly used. So the component of diazinon is the trade name. So the chemical component is chlorpyrifos. So that is why I had to settle on it. And it is an example of an organophosphate that is used in the eradication of ticks.
Okay, thank you. I'll call on Dr. Makwali. You may ask your question.
Oh, okay.
Thank you, Miriam, for your good presentation.
Thank you too.
When you're growing the isolates,
yes.
Why didn't you incubate 5 days?
Why did I?
Why did you not incubate beyond day 5?
Okay. From the literature, growth is between 0 to 5 days. Yeah, from the literature, that is what guided me. But even when we look at the degradation, even if the growth went up to day 5, you find that the optimum degradation is not even at day 5, it is at day 4. So the optimum is even 4. So 5 was like within the right range, the correct range to test for degradation.
If you, as per your figures,
yes,
the maximum, the optimal growth was on day 5.
Yeah, growth is day 5, but degradation. Okay, there were 2 ODs, but the first OD of 600 was testing growth.
Yes.
So growth like shows some lag phase, then there is maximum growth. So maximum growth was at day 5. That is OD 600. Then at OD 480, that is where I was testing biodegradability. And you realize that at OD 480, the maximum OD is recorded at day 4. So for growth, yes, 5 is maximum, but for degradation, 4 is the optimum.
Okay, thank you. I'll call on maybe 1 or 2 more questions from the floor.
Okay.
maybe I can ask, I'm assuming in soil, these isolates do not exist in isolation. So
Yeah.
would you, you didn't do it in your research, but would you expect to see if you were to create a mixture of these different isolates, do you expect to see some synergy, some antagonism? What do you think you would expect to see in terms of their ability to degrade the organophosphates? Thank you.
Yeah, thank you for the question. So the isolates surely do not exist in isolation. And I first of all utilize enrichment method that would knock out the non-degraders. So in the enrichment method, I used CP, so pyrophos, as the soil energy source, so that anything that doesn't utilize it cannot be able to grow, but as much as it is in the soil. That is first of all how I knocked out the non-degraders. Then for the synergies, that I think is one of my, that should be a recommendation for this study, whereby like the ones that could utilize similar optimum conditions can be put together and then observe the rate of biodegradation. It's a recommendation of the study. Should be a recommendation of the study. So the ones that work under similar conditions can be put together to also test degradability and see the rate of degradation. Like, is it also still maybe going to be a day 4? Is it going to be faster? Or maybe is it going to be less? Or maybe there is one that is going to antagonize the other. That is the next set of experiments that are supposed to be done after this. So this was supposed to pioneer so that now the rest of the experiments can come depending on the conditions that can be shared by most of the isolates.
Okay, thank you, Miriam.
Yeah.
Are there any other questions from anyone? Okay, so there are no questions, and I can also see there's no questions in the chat. So I'd like to appreciate all the presenters who presented in this session in the afternoon. I hope you've been able to take the questions and comments that have been made and incorporate them as you proceed with your project, if you're preparing for your thesis, to improve on it. And of course, like for Miriam, suggestions for further projects that you can pursue within your research. I hope that you will also join the other presentations going forward from tomorrow and Wednesday and Thursday. Other than that, I wish you a lovely evening. I bring this session to a close. Thank you. So you may leave at your own pleasure. Thank you.
Recording stopped.
most of these these methods have their own drawbacks. For instance, in steam distillation, it usually involves high heat, and we have thermal degradation. Because of this high heat, we degrade some of this essential oil. Also, the method usually takes longer times, and sometimes it usually causes the hydrolysis of some components. The organic solvent extraction is actually the widely used method, but the solvents being used, there are these organic solvents which are toxic. Some of them are toxic. Some of them, when released to the environment, they are not environmental benign. They're also expensive, taking note that they are petroleum product derived. And also, whenever these essential oils are extracted, these solvents, they cause contamination, because sometimes where these essentials are being used, less in food industry, we want no any trace of this solvent, but sometimes we are unable to remove all the solvent. So we have these methods with their shortcoming, and therefore we need an alternative method that can offer an immediate advantage over these traditional methods of extraction. Next slide. So when we talk about green extraction, we talk about the elimination or the use, the elimination and the use of generation of hazardous substances. And therefore my work involves green extraction, whereby I'm trying to come up with an alternative solvent which can eliminate the use and generation of these hazardous substances, especially the use of organic solvent in large amounts. So next slide. Now, what is supercritical fluids? If you look at the phase diagram, we usually have the solid, we have the liquid part, and then we have the gas. Now, whenever you are in this region here, on this boundary, we expect that at this boundary, we'll have your matter being in two phases, that is liquid and the gas. Some research done when the temperature was increased, and also the pressure, what happened when the temperature was increased, then what happened is we are moving along this boundary, and we expected that this boundary, it was expected that this boundary remains. But at one point, this boundary, as you can see on that visual aid on part 4, as you increase the temperature and pressure, at one point, that boundary disappeared. And actually, now it becomes one phase. And that point where that phase disappeared, that's what we call critical point. And anything beyond the critical point, that's what we call, or that region is what we call supercritical region. It's another phase whereby we have a matter having its own properties. And I'll talk about those properties. Like you'll find that the matter at this supercritical region will have liquid densities such that it will be diffusing, or it will have solubilities like liquid, and it has viscosity as gases. What that means is we have, or it diffuses as gas. So it has this dual characteristic. So anything beyond critical point usually have dual characteristics, whereby it's usually dissolve like a liquid and diffuse as a gas. Now, this is a good, and this is what we usually utilize in supercritical fluids, because when we talk about extraction, when you're extracting something, you know very well it has to penetrate through the matrix. Then it reaches to your analyte of interest, and then it has to dissolve it, and then transport it back to the bulk solvent. So when you talk about when you're extracting with a liquid, yes, liquid can actually have good solubilities, but it doesn't diffuse that well as compared to gas. But when we talk about the gas itself, it usually diffuses very well, but it has lower solubilities. But matter at this supercritical region, it has the dual characteristics such that it is diffusing like a gas, so it will diffuse into your matrix very well. Then when it reaches to your analyte of interest, it will be dissolving like a liquid. Then it becomes a super solvent, extracting solvent, which has both characteristics for a gas and a liquid. Next slide. Now, when you talk about supercritical fluids, any matter can go to supercritical or can get into supercritical region. For instance, if you look at this table, I've listed some of the I'm actually now from this table, you can see that I've listed some of the properties of some of these fluids, which can get into critical point. And you can see now water can be the best solvent, the most cleaner solvent. But if you see critical point for water, it's usually at 374 degrees Celsius and a pressure of 218 degrees Celsius. Therefore, if we have to use it, then we have to heat it at very high temperatures and also at higher pressures. Therefore, but when you look at carbon dioxide, its critical points is usually that 1 degree Celsius and 72 around 73 atmosphere for pressure. Therefore, it becomes ideal. Next slide. It becomes a very ideal supercritical fluid when it comes to extraction, and it's usually cheap and readily available. And for instance, for now, we are going to an era where we want to eradicate the use of petroleum products, and carbon dioxide is actually one of the byproduct of ethanol production. Therefore, as we are producing it in higher levels, as Dr. Moredi was stating it, we can reuse it to actually use it as a solvent. Also, when it comes to cleanup, because it is gas at a room temperature, therefore we don't need those post-extraction methods. We can just reduce the pressure and it just return to its gaseous form. Next slide. Now, this is how the supercritical fluid extractor looks like. We have the extracting vessel, then we have the packed carbon dioxide. Then we usually release the carbon dioxide, and within this extracting vessel, that's where we actually now regulate. We regulate our pressure and temperature to match that of critical points. That is around 32 degrees Celsius and 73 atmosphere. Then when we extract, then we collect. I'd love us to check here in collection. So here we can have very efficient way of collection, but if we don't optimize the collection part, so we might extract very well, but here we end up losing whatever we have extracted. And that's one. Next slide. And that's why we are talking about here. We know that the extraction process usually involves sample preparation, extraction, the collection, and analysis. The collection, when it comes to supercritical fluid extraction, is usually very important because here we are dealing with and especially when it comes to essential oil, we are dealing with volatile compounds. Therefore, we can end up extracting them very well, but end up losing them when collecting. Next slide. Yes, please.
Yes, reading the last once again.
Yes.
Yes, I'm being told that time can summarize so that we can.
Okay, fine. And that's exactly what I'll do. I'll just summarize. Actually, I think we are not very aware about what is supercritical fluid. So basically what we used or what we did was to evaluate. We modeled on the collection point. I've stated that the collection point is usually and very important when it comes to collection of volatile compounds. So we actually modeled. Next slide. Next slide. Next slide. Next slide. Next slide, please. So basically what we did, we modeled the collection point, And we actually established what are because there are so many factors which affects the collection point. We modeled those factors. We were able to actually establish which are the most important factors, the most significant, and then using screening models. And then we used optimization models to come up with the most optimum conditions for the collection. Then after that, we utilize that to extract and analyze using the GC. Then next slide. Next slide, please. Next slide. So after determining those collection models, we used Pareto charts, and we were able to actually now determine which were more significant, which were not, which were less significant. And then next slide. With that, we were able to actually see the most significant variables were the time, solvent temperature, and flow rates. And then yes, thank you. Now, next slide, please. After that, we modeled for optimization, for the extraction of optimization. And we actually checked on, next slide. Next slide, please. Next slide. So we were able to check or to evaluate the model fitness, and we were able to see the values, the predicted values and the actual values we got. So the models were very okay. And from there now, we actually utilize that method for extraction of essential oil. Next slide. Next slide. So these are the plants which we worked on. We worked on chamomile plants. Next slide. We worked also in a rabbit brush. Next slide. Then on a skunk brush. Next slide. And we extracted and evaluated our results using GCMS. And these are some of the results which we got. Next slide. And the summary for that, we are able to actually quantitatively, because when we talk about quantitatively, it means that we were able to establish the actual amount of from the mass we took for that plant, the actual amount of the essential oil present. And in rabbit brush, we were able to realize that limonene was the highest as compared to the rest. And it had more of monoterpenes. As in my introduction, we talked about different types of essential oils. For rabbit brush, it was more of monoterpenes. Then next slide. Next slide. And the chamomile. We can see that from the different plants, we're able to establish if they were containing more of monoterpenes or sequiterpenes or oxygenated derivatives. Next slide. So from my work, I wish I could have been able, just because of time we had to rush, but we did modeling, and actually we are able to actually prove that design of experiment approach can be used to model the collection point when it comes to supercritical fluid extraction. And then we also established that most of the time we usually optimize using, like considering one variable and running other variables at constant. But this time around, we were able to prove that interaction variables. So when we do that, we don't usually optimize our process. But when we do, or we work with the models, we can be able to actually get even the results for the interaction of the different factors which are affecting our processes. And we were able to establish that different plants from different climatic conditions and also from different families, they usually contains different essential oils, and we were able to actually come up with a method for quantitative, when the underlined word is quantitative. Most of these extraction methods, they don't usually give quantitative results, but you can use them for qualitative results, but for quantitative, it's usually limited. Next slide. So I love to thank or to acknowledge my university, also the University of Eldoret, for organizing this wonderful conference to be able to showcase what we are doing. And I'll also like to acknowledge my green chemistry lab where I did this work. So thank you very much, and thank you for listening to me.
Thank you, Dr. That was a wonderful presentation. So briefly, from the members, do you have any questions or any comments to us, our presenter? Any question or comment from the members? Well, I think we'll proceed. Thank you, Dr. for the wonderful presentation. We really appreciate. And
yes, thank you, Chair.
Karibu sana. So that marks the end of our presentation in this session. So at this time, I will hand over the meeting to the chair of the conference, Dr. Deli Sant.
Thank you, Mr. Kamal. So members, I think we've come to the end of day one of our Chemical Sciences Conference. And I will want to take this opportunity to thank each and every one of us. Okay. I take this opportunity to thank each and every one of us for your participation. For the participants who are presenting their work, I want to say thank you for choosing to present your research findings in this forum. And then for the chairs and the rapporteurs of the various sessions, we thank all of you because it has actually been smooth. And I can say that the day has been a success. So before we call it a day, I'll just take you through a brief of what we expect tomorrow, because the program is a bit different from what we had today. So tomorrow we begin at 8. Okay, so we'll have 8:20, we'll have registration, after which we'll have the national anthem, East African anthem, and a prayer. And then the parallel sessions begin at 8:30. Okay, 8:30, we'll be in room 1 and room 2, 8:30 to 10:30. Okay, after that, we'll have a break. And then at 10:55, we all log in to the main conference hall. Now in the main conference hall, we'll have 2 talks. We'll have the VC presenting her talk from 11 o'clock. And then after the VC, we'll have Professor Christophe from Belgium presenting his talk. And then we'll break for lunch after the 2 talks and come back for the afternoon session at 1:55. In the afternoon, we'll have 1 keynote speaker. That's our Professor Lusweti Kitui. So between 2 o'clock and 3 o'clock, we'll have the keynote speech, after which again we break into the 2 rooms, room 1, room 2 for the parallel sessions. And then we expect to end around 4:20. So members, that is a brief of what we expect tomorrow. So it's a bit different from what we had today. So hope you take a note, and of course you have your programs which you can refer to. So once again, I want to say thank you and hope to see all of us tomorrow again. Thank you.
You can request him. We will request
The University of Eldoret is a modern university situated approximately 9 kilometers northeast of Eldoret town in Uasin Gishu County, Kenya. Founded in 1946 and converted to a teachers training college in 1984, in 1990 it became Chepkoilel campus of Moi University, offering natural, basic and applied science programs. In August 2010, the campus was upgraded to Chepkoilel University College, a constituent college of Moi University. Upon the award of charter by the President on 11th February 2013, the University College was renamed University of Eldoret. Our vision is a premier university that nurtures global leaders and innovators. Our mission is to provide quality education, training, research and consultancy in science, agriculture and technology to meet the needs and aspirations of a dynamic society.